And again, Bruce Beutler described this, and he did win the Nobel Prize. So we thought this was incredibly interesting, and actually this article is the second most quoted article in hepatology in the last quarter of a century. So this is showing that there is an inflammatory mediator that can cause liver injury. But there were a lot of questions that still remain to be answered. So what was driving the TNF production, and why was it overproduced in people with alcoholic hepatitis? So if we fast forward 25 years later, where are we now? So I'm going to give you first an overview of the gut barrier. And I'm talking about gut barrier and not gut flora, because gut flora is only one component. So you have the bacteria, these little blue things up here. Then you have the mucin, this green junk that separates the bacteria from the epithelial surface, and that's very, very important because you don't want all that bacteria sitting right on that epithelial surface. So here are the epithelial cells in purple. You've got specialized immune cells called the Panna cells that release peptides that regulate the bacterial flora. You've got these red things, the tight junctions. You've got the goblet cells that secrete mucus. You've got the dendritic cells. And so the bottom line, a screw up in any one of these things can cause gut barrier dysfunction. And I'm just showing you an example of one of these particular things, which are the tight junctions. And there are tons of different proteins that make up the tight junctions. And so a mess up in any one of these things can cause a problem. And so I'll show you, we actually measure all these in our laboratory. Here's an electron micrograph. You can kind of visualize how endotoxin up here might scamper across this tight junction here, get into the portal circulation, the superhighway, go into the liver, and activate Cooper cells. So as a gastroenterologist, an old gastroenterologist, typically you think about the stomach making acid, the small bowel absorbing nutrients, the colon for fine regulation of water absorption. But actually we have an increasing gradient of bacteria in our GI tract also. And very importantly, there are many more bacteria in our GI tract than we have cells in our body. There are many more genes in the bacteria than we have genes in our body. So realistically, we're just a receptacle for our bacteria. And the bacteria differ dramatically from one body surface to another. So if you look at phyla in the gut, why it's mainly the Firmicutes and Bacteroides. It's actually very different just in the mouth, you can see, and highly different from the vagina. So we measure this bacteria not by culturing, but by a thing called 16S ribosomal RNA. And so a lot of groups focus on measuring bacteria. We focus on measuring what the bacteria make. So this is called metagenomics. We focus on metabolomics because we're actually interested in what the bacteria are doing. And this is a cartoon actually from another group study, but just showing giving short-term antibiotics will markedly alter your bacterial metabolites. So there's a decrease in short-chain fatty acid, branched-chain amino acids, and an alteration in bile acids. And I'm going to come back to that when we talk about our studies in alcoholic liver disease. Now, why do we care about all this? Why do the clinicians in the audience worry about this? Well, we got Jason sitting there, but unfortunately, Jason, cancer is not the biggest problem in the country. It's obesity, and especially in Kentucky. And so 2/3 of adults are overweight or obese, and actually 1/3 of people have NAFL. So Jesse harasses me about going to clinic, seeing patients. I could be in clinic 24 hours a day, 20 days a week, seeing patients with fatty liver disease. Big, big problem. Now, in another life, I told you that I do nutrition research, and I'm actually a nutritionist. And when I used to talk about obesity, it was a simple thing. So it's calories in, calories out, energy in, energy out. And nobody was interested in other factors. But when I go to clinic, this is what I hear all day. You know, I eat like a bird and I still gain weight. So are all my patients lying to me or not? Now, I gave this talk in Tampa, a variation of it, and the guy came up to me afterwards and he said, you know, birds actually eat a whole lot. So, as everybody knows who writes a grant, you have to have an alternative strategy. So we came up with an alternative strategy, and that was you can blame your gut flora here for obesity and fatty liver disease. And you can blame your mom. So that's where you get your gut bacteria from. And the talk that I gave at AGA was actually right around Mother's Day, so this wasn't the popular slide. But this whole thing is important because of the concept of low-grade inflammation. So this is a cartoon from our group showing that we think you get metabolic endotoxemia when you get altered gut flora, increased gut permeability, bacterial toxins leak across, they activate these toll receptors, you get increased cytokine production, liver injury, and systemic inflammation. And this has been our cartoon for alcoholic liver disease. Now, this is a cartoon from Canyon coworkers who are the leaders in Europe in obesity. And they say basically exactly the same thing. And that this low-grade inflammation is responsible basically for all of the complications of obesity, including fatty liver. So, moving on to then how we get there. So we'll talk about high fructose and high fat diets to begin with. And this is a good place to talk about this, that many of these companies are housed in Louisville, like KFC, Papa John's. And in the laboratory, we feed animals either a high fat diet or a high fructose diet to make them overweight and get fatty liver. Now, interest in this was stimulated by work from Washington University about 10 years ago when they showed that germ-free animals had less body fat, but greater food consumption and less energy expenditure than conventional animals or germ-free animals that were then basically given a stool transplant. So if you had a pill for this, you could cure obesity, you'd be a zillionaire. Then other groups showed that genetically obese mice, so BOB and DBDB animals, had increased gut permeability as assessed by horseradish peroxidase compared to wild type, and increased endotoxin in their bloodstream. Now, a group that we've worked with, Dr. Wenke Feng generated that collaboration, showed that if you just feed regular animals a high-fat diet, you can measure bacterial DNA in their bloodstream at a week, and it's even more at 4 weeks. And if you take out their mesenteric adipose tissue or lymph nodes, it actually goes to the adipose tissue first. And so this is important because it highlights the concept that visceral fat isn't just this glob of stuff. It's highly bioactive, and it's making inflammatory cytokines. It's doing bad things. And then the article that probably everybody has either seen or heard about, again from WashU, they took genetically obese mice, and they showed that they had an altered gut flora with a low level of Bacteroides, high level of Firmicutes, compared to the wild type mice. And then what they did is they took the stool from the wild type or the obese animals and did transplants into germ-free animals. And the ones that got the stool from the obese mice gained more body fat. Now, an article that I like even better, because this is what I do for a living and see for a living all the time, is we put animals on a high-fat diet. Got 10 animals, 8 of them gain a fair amount of weight. There's one of them that's a monster, and there's another one that nothing happens to. And so this is just a variation study. And so they took the one that didn't gain weight, and they took the monster, and they took stool from them and did basic transplants. So this is the rapid responder and the non-responder. So here's their liver histology. You can see the terrible fatty liver in the rapid responder. Here's the HOMA for insulin resistance, and here's measuring fat in the liver. So basically, your phenotype can be determined by a stool transplant. So this is highly important. You don't have to worry about animals. So a lot of you know Ryan Hurt, who was here and now at the Mayo Clinic. So Ryan actually took Super Size Me, the movie, froze the frame so he could look at the medical record of the guy. And so here's baseline, 18 and 21 days. And his doctor forced him to quit this at the end of this study because his liver enzymes were going up actually dramatically. And so he had severe NASH. Now, the thing that our group is focused on has been fructose. And part of the reason for this is it's easy to deal with it in a way. Just tell people to quit drinking pot. So that's the simple thing to do. I like simple solutions. So a lot of this work was done by Nina Bergheim when she was a postdoc. Miriam Voss was a pediatric GI fellow. She's now at Emory and works both with Emory and the CDC, and is actually the leader in the country in pediatric NAFL, and Ming Song, who's part of our group. And the first thing that we did was just feed animals regular water or give them artificial sweetener, glucose, sucrose, or fructose. And then we cut their bodies open. And you can see all the visceral fat here with the glucose and sucrose, less with the fructose. And that's because the fat's in the liver. And that's shown here with fructose having the highest amount of hepatic fat, the highest amount of this bad lipid peroxidation product, 4-hydroxynonenal, a highly toxic aldehyde. And fructose caused the greatest amount of endotoxemia and the highest amount of cleanup in the liver. Then we went on to show that if we gave these animals a non-absorbable antibiotic, alter their gut flora, we would totally protect against this endotoxemia and markedly reduce the fatty liver, showing the importance of gut flora and fructose-induced obesity and fatty liver disease. And we've done a lot of other studies in this that are mechanistic that I'm not going to bore you with. Then Dr. Song from our group asked, what if you have two common nutritional alterations? So marginal copper deficiency is actually fairly frequent in the United States, and we know that a lot of people drink fructose. So, and there was old literature from the 1970s saying a high amount of fructose worsened copper deficiency. So we looked at this in experimental animals, and if you just look over here, the open circles are the fatty liver in the fructose-fed marginal copper deficient diet. And they were the ones that had the highest ALT levels. They also got a lot of iron in their liver that I won't go into the mechanisms for. But basically, I see this every week in clinic. So patients are sent in to me with increased liver enzymes. Half the time, their doctor thinks they're brilliant because they think they've diagnosed Wilson's disease because the patient has a low serum copper. The other half think they're brilliant because the guy, they think he has hemochromatosis because he has an increased iron. And what they're really doing is drinking 12 cans of Mountain Dew a day, and when they stop that, everything gets better. Now, in the nutrition area, you'd think that these guys would be nice guys, but they actually are very passionate about some things and really almost get into fistfights. And one of them is actually whether it's just sugar that's bad or whether it's fructose. And the corn people are really very serious about protecting fructose. So this is an incredibly neat study where they fed a high glucose diet to wild-type animals, and you can see this big liver here, and then knock out animals that couldn't convert glucose to fructose, and everything's normal. And here you can see the fatty liver and the wild type to knock out no fatty liver. And if you just focus over here, here's the liver enzyme in the wild type and the knockout animal. And this particular enzyme is aldose reductase. This is the enzyme that Dr. Bhatnagar has spent his life working on. And so we think that this is actually a very important observation. And so it's not only the fructose that you take in exogenously, but it's what you make endogenously. So basically, high fructose could explain all of the complications of the metabolic syndrome. So I'll switch over now to alcohol and a high-fat diet and their interactions. And this work was done by Irina Kirpik in our group. And it's very similar to the obesity story. So you have a leaky gut, and the toxin goes across, activates Cooper cells, get TNF production, liver injury. And we've shown that if you culture intestinal epithelial cells with alcohol, you get a decrease in epithelial resistance, so those tight junctions fall apart. And this is work that we just recently did with the NIH. So we took blood from people that came into an alcohol treatment program, so not with liver disease, and showed that they had endotoxemia that slowly decreased during their hospital course. Now, we've talked about the fructose epidemic. There's also the linoleic acid epidemic, a certain type of fat. It's actually in corn oil. So in one of my talks, I said our solution to the health problem is bomb Iowa, get rid of everything. That wasn't popular. But you can see PUFA intake and linoleic acid intake has increased dramatically over the last century. And there are epidemiologic studies showing that in humans that the cirrhotics, alcoholic cirrhotics, tend to have more linoleic acid in their diet. And then there were early studies suggesting that you had to have this unsaturated fat in your diet to get alcoholic liver disease, and that saturated fat would protect. So we looked into that more. And so if you go back, remember we talked about tight junctions. So this is zonula occlusion one of the tight junctions. So here's our unsaturated fat diet alone, and then with alcohol, a marked reduction. If you look at the saturated fat, basically normal. Then endotoxin leaks across in the unsaturated fat with alcohol, not the saturated fat. If you look at liver injury, with the unsaturated fat, you get injury. The same amount of alcohol with the saturated fat, no liver injury. And here's liver triglycerides for comparison, and ALT for comparison. So very recently, then we've gone on to look and see, does this have anything to do with gut flora? So we said that there are two big phyla in humans and in animals. So it's the Bacteroides, this kind of brown chunk here, and the Firmicutes in green. So you can see in the saturated fat with alcohol over time, nothing really happens. But here, if you look at the unsaturated fat with alcohol, at a week, not much is going on. 6 weeks, you start to see a little bit coming in at the edges here. At 8 weeks, you have two new phyla coming in here, the Proteobacter and the Actinobacter. And so we think this is causing major changes in the metabolites that these bacteria are making and causing liver injury. So how could the linoleic acid actually be inducing liver injury? Well, linoleic acid gets metabolized over this lipoxygenase pathway to things called CODIs. Those are oxidized products of linoleic acid. We call them oxalams. They're very toxic and reactive. And very recently, we've shown that they bind a receptor called TRPV1, which is very important for neuroinflammation. And we've shown that this activates the inflammasome, which causes IL-1 production. So that takes us all the way back to our original research with IL-1 25, 30 years ago. In our intestinal epithelial cells, we showed when we added these HODIs in, especially the 13 HODIs with alcohol, it just totally knocked out epithelial resistance. And in our liver cells, it's screwed up mitochondrial function. So mechanistically, these are different ways that can be causing toxicity. We then showed that this TRPV1, this inflammatory receptor, was upregulated in the liver with alcohol. And when we used knockout animals and fed them alcohol, you can see they have much slower liver enzymes than the wild type. So this was protective. So in summary, PUFAs, but it's the linoleic acid, the N6, that causes alcohol-related hepatotoxicity, saturated fat protects, and we think these oxalams are partially responsible. So the third type of liver injury that I'm going to touch on, just briefly, is toxicant-associated steatohepatitis. And we actually described that in 2010. McClain was the lead author on that in Hepatology. And what we were doing, this is Rubbertown in Louisville, and we have a long-standing relationship with industries there. And we went back and looked at patients who had had liver biopsies in the 1970s. We had a surveillance program for liver cancer. And so the liver biopsies in the patients who didn't get cancer all showed, or 80% of them showed steatohepatitis. Yet these patients didn't drink and they weren't overweight. And so we thought it was their exposure to a toxin in that industry called vinyl chloride that was causing this. And this was probably the most unsuccessful surveillance program known to man. So we did everything right except we had the wrong biomarkers. So we were looking for abnormal liver enzymes. And so here, AST and ALT, healthy unexposed people. Here are healthy chemical workers. Here are people with biopsy documented bad inflammation. Normal AST and ALT. So now we've been able to go back and see, is there a better biomarker? And we have this thing, cytokeratin 18. We also looked at these pro-inflammatory cytokines like TNF, IL-1, and IL-8. They were all elevated, just like we see in alcoholic hepatitis, and the antioxidant capacity was very reduced. So biochemically, they look just like alcoholic hepatitis. We then went on to do this fancy metabolomics thing, and I don't want you to read this, but just know that up here, the most abnormal things were these heats and HODIs that were also elevated, we said, in alcoholic liver disease, and they're also elevated in just obesity-related liver disease. So here we have a common denominator between three types of steatohepatitis. Now, Gavin Artiel and several other groups, people from our group, Bert Watson, and Julianne Artiel are looking at arsenic as it relates to liver disease. And arsenic's a hot topic. If you have fracking, they're worried about it getting in the water supply. And so they asked whether if you had a fatty liver due to a high fat diet, a Western diet, and then exposure to arsenic, would that cause liver injury? And indeed, that's exactly what they showed. So here's the combination of the two, and we're looking at ALT, we're looking at the bad cytokine tumor necrosis factor, we're looking at a thing that causes breakdown of fibrin by 1, and here's the good cytokine interleukin-10, which is reduced. Now, arsenic can do a lot of different things. It screws up your tight junctions. It's toxic to certain gut bacteria. And Gavin, our teal, is focused on hepatic fibrin. And so here we can see arsenic plus a high-fat diet has much more fibrin. And then he wanted to see, could he give a prebiotic to attenuate that? And so that's what he's done here. So instead of giving a probiotic, it's a prebiotic, fructose, and you can see that that's much improved. So I'm going to spend the final part of my talk about nutritional intervention. And I like nutrition as an intervention because you don't have to go through long FDA approval type things. It's going to take 10 years to get a drug on a market. So if you can find some nutritional intervention, you can do it quickly. You won't make nearly as much money on it, but it's an intervention that will come to the market quickly. So I'm going to look at a trace metal, a fat, and a probiotic. And we're working on all three of these. So the zinc studies were done by Kurt Watson, Matt Cave, and Dr. Zoe, who's now at North Carolina. And actually, Matt Cave is just finishing up his clinical trial on this with really exciting results in alcoholic cirrhotics. But zinc has antioxidant function. The thing that we're going to focus on is it's a critical part of zinc finger protein. So those are critical proteins that do a whole lot of things in the body that are vital for normal function. And these are early studies that I did back in the 70s, again showing that people with alcoholic liver disease have low serum zinc levels, and those with cirrhosis have the lowest levels. They can have classic signs of zinc deficiency. Here are the skin lesions again. And there are many reasons for this. So there's a decreased intake. Beer doesn't have much zinc in it. Poor absorption, increased losses. But the important thing that I wasn't smart enough to figure out, but Dr. Zoe was, is that with oxidative stress, which you have in liver disease, the cysteines that hold this zinc in here get oxidatively modified, and the zinc leaks out, and you lose that zinc finger function then. And so we've been giving zinc to animals with experimental alcoholic liver disease. And so here's the one animal is getting alcohol, alcohol plus zinc. Alcohol, alcohol plus zinc. So you can see endotoxin levels are improved, serum TNF's improved. Any marker of oxidative stress here, if we look at 4-hydroxynonenal going across here, the brown junk is bad with alcohol, much less brown junk there with the zinc. So it looks much better. And if you just focus on this one, zinc finger protein here, HNF4 alpha, the activity goes down with alcohol, and it's basically totally corrected with zinc. And then the proof's in the pudding that you can see the liver enzymes are improved compared with zinc and alcohol compared to zinc alone, and much improved liver histology. And here's the animals not getting the zinc. So basically, zinc works at every level that we think liver injury occurs at. So that's why basically the gastroenterologists have this in their water supply for our liver patients. So we talked about a trace metal, we'll talk about a lipid. And the one that we're going to focus on is butyrate, a short-chain fatty acid. Now, why did we get into this? Well, we did this metabolomic stuff again that we talked about. So we take the stool, we give it to this chemist over on main campus. It's a genius, and I don't understand anything he does. And he gives us results back and publishes a paper a week later. So this has been a great relationship. And so what he showed was in the stool of the alcohol-fed animals, there's a decrease in short-chain fatty acids, especially butyrate, altered bile acids, and decreased branched-chain amino acids. Basically the same thing I showed you with the short-term antibiotic treatment earlier. And so we wondered, if you just gave that short-chain fatty acid back, that's low. Could you protect against alcoholic liver disease? And we picked on butyrate because it has epigenetic influences. So it's what's called an HDAC inhibitor. So it has huge influences on your epigenetics, which can change very quickly. And so here are endotoxin levels in alcohol-fed animals, and here they are with the prodrug butyrate called tributrin. Correctly, here's the ALT, and here it is with tributrin. So this is very exciting, and again, we're pursuing that further and looking at the epigenetic effects. Now, we try and do this systems biology approach when we're looking at organ entry due to alcohol. And one of the first things that we found actually several years ago with gene profiling was a decrease in a thing called intestinal trefoil factor. So it's kind of like fine wine, let that sit around. I should have jumped on that right away, because this was 50-fold reduction. You never see that. But fortunately, there was a guy, Winky Feng, that came to our group who was interested in hypoxia-inducible factor, and which is a critical transcriptional regulator. And he knew that ITF was regulated by HIP. And he also had an idea that probiotics may affect the HIP. And so we asked whether a certain probiotic, and we got lucky, we picked this Lactobacillus GG, would be a therapeutic option for alcoholic liver disease. And this is a unique paradigm. So we fed animals their alcohol diet for 6 weeks. Then we started them on their probiotics and treated them for 2 weeks while continuing the alcohol. So most other studies, when they do this, they start giving the probiotics over here, and so it's a prevention arm. This is like real-world treatment. And and for everybody, probiotics are live microorganisms that when you give them to a human, you get a beneficial result. So Dr. Feng was right. ITF was markedly reduced, HIP was reduced in our alcohol-fed animals in the intestine. And very importantly, he showed that LGG, and I'm not going to go through and show you all the data, but the broth that we cultured the LGG and the supernate basically was totally effective. So here is endotoxin leaking across an alcohol. Here's alcohol plus LGG. Here we give E. coli that are labeled with a fluorescent probe. You can take out the mesenteric adipose tissue that we talked about. Here are the alcohol-fed animals, a lot of bacteria migrating across, but none basically in the ones that get the LGG. If we look at hepatic TNF, up here, down with the LGG, marker of neutrophil infiltration up, down with LGG, liver enzymes improved. Here's histology down here, alcohol plus LGG. As I say, very importantly, he's shown that you don't need the live bacteria. You can use the supernate. So this is very important for patients with HIV that we may want to correct barrier function, people with immune dysfunction, transplant patients. You can give the supernate that has the active ingredient in it, and that's what we're working on. We also showed that there was a change in the gut flora. So this is kind of complicated, but I'll walk you through it. We have these two types of bacterial phyla again, the Bacteroides in brown and the Firmicutes in the light blue here. So this is pair-fed and alcohol at time 1, pair-fed and alcohol at time 2, pretty much the same. Here's 8 weeks. Here's the pair-fed, here's the alcohol. Again, we see green and red encroaching. When he gives the LGG, you get rid of the red and green, but you still have an altered ratio here with lower Bacteroides, more Firmicutes. If we look at this in more detail, later in the course, a bacteria takes off that loves to grow in an alkaline environment called alkaline genes. And indeed, the pH goes up in the alcohol-fed animals. So remember we said we had a decrease in the short-chain fatty acids, so the things that cause the pH to stay down, you're not making as much of. And as soon as we give the LGG here, the pH comes down. So back when I was doing the hepatic encephalopathy studies in the 1970s, we were studying this new drug lactulose. That would be called a prebiotic now. And one of the ways that we documented whether the people were taking it is we looked at their stool pH to see if their stool pH was dropping. By metabolomics, we showed that the LGG increased succinic acid, which positively regulates HIP. So this goes back to our whole metabolic pathway again. So in conclusion, there's extensive crosstalk between food, alcohol, toxins, and our gut microbiome, and probably more importantly, what the gut bacteria are making. Gut barrier dysfunction plays a critical role in fatty liver and steatohepatitis. And like I say, I could be talking and say it plays a big role in diabetes, plays a big role in rheumatoid arthritis, possibly in Alzheimer's and Parkinson's. There are a huge number of disease processes where gut barrier dysfunction is certainly associated. It provides multiple prevention and treatment targets. And so, again, there are quick ways of intervening here, but unfortunately, this is likely to be highly individual-specific and disease-specific. So this isn't going to be a one-size-fits-all. So let me stop and see if there are any questions. Oh, I need to.
Greg, there are people in the audience who wonder how do you talked about your mentor that got you to do some things.
Right.
Were you that not busy that you had time to do this? So there are people in the audience who are wondering, how do you develop a career of 30 years of being a physician's son? How do you start there?
Well, I mean, this was in my fellowship, so we had to do some research. And so the great thing, if you have, any little junk I cooked there, he would always say great. And he was just a fantastic guy. He was a guy who, when people stole his ideas, he was thrilled because he had so many ideas that he was just glad somebody else was doing them. So good mentors are great people.
So part of the process is really having everyone to go through research and then finding out who loves it and who doesn't.
Right.
Okay. Questions? Questions? Yes, sir.
So I was perplexed. Your talk number one, the fructose diet versus the glucose diet.
Right.
I've never seen that data that the fructose causes all the fatty deposition in the liver, and I just wondered
Jason, you need to get out of laboratory.
Yeah.
Read the New York Times.
I don't watch TV and I don't watch. So is it that the reductase is really overexpressed in the liver and that's why it happens, or what's the mechanism for the differential utilization of the fructose? Because if you don't have phosphorylated fructose, obviously you're not going to be able to use it.
So are you asking?
Repeat a little bit of the questions.
So I'm asking, why does the liver take the fructose and convert it to lipids as opposed to the adipocytes?
Well, so you do get obese, so the adipocytes do too. So I was just focusing on the liver there. So you will get visceral adiposity, so you'll get the same picture as you will with a high-fat diet.
Yeah, I saw you just showed that one slide where there was glucose and then there was fructose.
Yes, so why does your liver get to be a bigger target?
Right,
so,
right.
Yeah, and people aren't really sure of that yet. It's, but it is absolutely clear that there's a gradation of bad fat. So subcutaneous fat doesn't look good, but it's much less toxic than visceral fat, which is still much less toxic than hepatic fat. So, you know, cardiologists really work for us that heart attacks start in the liver.
One of the connections between visceral fat and hepatic steatosis is supposed to be the enzyme HSD1, which converts cortisone to cortisol, and then the liver is exposed to very large amounts of cortisol, which have a lot of negative effects on lipid and glucose metabolism. Have you looked at that connection as primary, perhaps adipocyte in the intestine being the primary target for some of your model interventions, and the liver being sort of a secondary response to the effects on the small bowel?
Yeah, so that's a good question. The answer is no, we haven't looked at it, but it's a good hypothesis.
I saw a question here, and then we're going to go here.
I had a question, because I like your comment about I eat like a bird and I still gain weight. I see that a lot in actually cancer survivors who say that after their chemotherapy they continue to gain weight even if they're not on hormonal antagonists, and without a good reason. Has anyone looked into like the gut barrier changes that may occur with cancer survivors?
No, but certainly their gut flora could be altered, and cancer chemotherapeutic agents obviously are incredibly toxic. So they could have altered gut flora and maybe permanently done it or done something to gut barrier function.
I thought it was wonderful pathophysiology data. I was wondering if much has made its way up to clinical observations.
Yeah.
Should we be adding zinc to our bourbon, for instance? Or?
Actually, the zinc would be one thing. What we'd like to do with the alcohol industry is actually figure out what's in the LBG and have that added in there. So, I mean, it's a fermentation process for alcohol anyway. So if you were fermenting and you had the right thing in there, why, you might be able to prevent development of alcoholic liver disease. So that's one of the things that we're interested in. There's a lot of use of zinc around the country for alcoholic and non-alcoholic steatohepatitis. Some things that we didn't get into related to cytokines. Pentoxifylline is something that we use. It's a phosphodiesterase inhibitor. I drink 20 cups of coffee a day, so that's what coffee is too. It's a wheat phosphodiesterase inhibitor, so that's why coffee is good for you and for your liver.
I don't want to see his gut flora.
Great talk, but I wonder, do you know whether alcohol could possibly be a genetic modifier, inducing a derepressor of an enzyme system through genes in order to act on things like the interleukin 1 and TNF alpha?
Yeah, so the question is what alcohol does to cause increased interleukin 1 and TNF production, and that those things are multifactorial, that you get iron overload in macrophages that influence that. You get increased NF-kappa B, as I actually showed there, but I didn't get into it. So there are at least 4 or 5 things that are well documented, and people are still studying why that happens.
Greg, upstream of a lot of you presented, you talked about reactive oxygen species and low glutathione and things like that.
Okay.
And antioxidants haven't been that helpful.
So actually that's...
Are we trying the wrong approach for that?
So that's the talk that I'm giving Saturday at the liver meetings, is ASH and NASH comparisons. And so, and that's the first thing that people probably looked at in both these disease processes was oxidative stress. And for non-alcoholic steatohepatitis, our first line of therapy is vitamin E, 800 international units a day. So that will get fat out of the liver. That will make your liver enzymes come down to normal. And so we give that to all of our patients who have fatty liver. What it won't do is it won't reverse fibrosis. So once you're at stage 3 or something like that, it's probably going to be less effective. But you're absolutely right. In alcoholic hepatitis, which is more severe inflammation, probably, why the antioxidants haven't worked. And as you know, maybe we're doing the wrong thing. Maybe we should be inducing our own body's antioxidants systems instead of giving exogenous antioxidants. So that's a huge debate.
One last question back there.
I have again a question about fructose and glucose difference. So, what if you take sucrose then? It will be broken down into glucose and fructose.
Right. So it's a 50-50 mixture. Yeah. So you kind of get what you would expect. So if you give glucose long enough, you will get fatty liver. If you give sucrose a little less long, you'll get fatty liver. If you give fructose, you don't have to give it near as long to get fatty liver.
Well, thank you, everyone. And highlights why Louisville has one of the strongest nutrition GI hepatology programs in the country, thanks to this guy and a lot of other people in the audience.